Ready. Okay. Hi, everyone, and welcome to Mountain Talks. I am Lindsay Drake, and I am here with P.C. Rawson, Assistant Professor of Chemical and Biomolecular Engineering, Angela Brown, who is going to give a brief talk about 10-15 minutes regarding her research for combating antibiotic resistance. After her presentation, we will take questions via the chat box, and I will moderate that portion. I'll read your question back to Angela so that she can answer it. And we'll do that for about 20 to 30 minutes, and then we'll conclude the presentation. So at this point, I will turn everything over to Angela.
All right. Thank you. So today, what I'm going to talk about are really the factors that have led to antibiotic resistance, and then what we're working on in my lab to combat those. So first, I wanted to start with some statistics from the World Health Organization. So this shows the number of deaths in high, middle, and low-income countries due to a number of different factors. And the gray and the white bars at the very bottom of each of these graphs shows deaths from infectious diseases. And so you can see that in low-income countries and middle-income countries, death by infectious disease is very prevalent. But what I think is really hopeful about this graph is that you can see that the number of deaths due to infectious diseases is decreasing. And that's projected to continue through 2030. The reason for that decrease is what's been described as a four-walled fort. So Carl Nathan is a professor at Cornell who's described this four walls of this fort. So the four walls would be sanitation. So we have access to clean drinking water now. Doctors are very careful about washing their hands before they treat you, those sorts of things. Nutrition, we have access to healthier foods than we used to. Immunization, so a lot of these diseases that used to kill people, we now have a simple shot that can protect you for a lifetime. We don't even think about these diseases anymore. And then antibiotics. So if you do get sick from a bacterial illness, we have ways to treat that. So in this paper, Carl Nathan makes this argument that the fourth wall is crumbling, and this has been in the news. The antibiotic resistance is a major problem, and I would say the third wall is a little bit shaky. There's an anti-immunization movement out there, and I think that's a really scary prospect. That really what we're left with then is just sanitation and nutrition for combating diseases, and so clearly we need something new. So the reason for antibiotic resistance, there's several factors that have led to this point. This graph shows when a particular antibiotic was introduced. The up arrows show when it was introduced. The down arrows show when resistance was first discovered. And so you can see back in the 1940s, 1950s, even the 60s, after an antibiotic was introduced, there was probably a good 10, 15 years before resistance was discovered. But then if you look over towards the 1990s and 2000s, a lot of times an antibiotic was introduced, and then within a year or two, resistance was discovered. And there's a lot of factors leading to this. One would be the overuse and misuse of antibiotics. Another factor would be that we haven't actually discovered any new classes of antibiotics. And so what we're doing nowadays is sort of chemically modifying existing antibiotics. And so resistance can occur a little bit faster for that reason. And that is shown in this graph here. So you can see that in the 1940s and 50s and the 60s even, this was called the golden age of antibiotic discovery. But since the 1980s, we haven't discovered any new classes of antibiotics. So there have been new antibiotics put to market, but those, like I mentioned, are just chemically modified versions of what we already have, or combination drugs. Another factor that leads into the difficulty in developing antibiotics is that there's only so many targets. So to develop an antibiotic, you really need two things. First, that target has to be essential for the bacteria to live, because you're trying to kill the bacteria. And then that target also has to be different than the corresponding version in the human, otherwise there would be really nasty side effects. And so most of the antibiotics that are out there target one of three targets: cell wall, protein synthesis, or DNA replication and repair. And so there are people that are trying to uncover new targets for the development of more antibiotics. Another issue is that gram-negative bacteria are really difficult to treat. And this is because of their structure. So those types of bacteria have two membranes. And so it's really difficult to get an antibiotic into the bacterial cell. So if your target is inside the cell, the antibiotic has to get in there, and it's just really difficult to do that with these two membranes of the gram-negative bacteria. So what some people have done, including my lab, is to focus on these molecules called virulence factors. So these are molecules that are produced just by pathogenic bacteria that give them an advantage in establishing an infection in the body. And so I have, in this cartoon, I have a couple shown, but there's many more than this. But this is just to give you an idea of what these virulence factors could be. Bacteria might produce adhesins or other types of molecules that allow them to bind to a surface that they couldn't otherwise bind to. They can produce effectors and enzymes to maybe get nutrients that they need to survive. And then what we focus on in my lab are toxins, which the bacteria secrete to kill local cells. I'm sorry, I'm going to take a step back. So the idea in our lab is that if we could understand the mechanism that these toxins use to kill the cell, we could block those mechanisms. And then the bacteria will be cleared through natural immune systems. So in this case, we're not actually trying to kill the bacteria, we're just setting up the conditions that the immune system would be able to clear the infection more readily. So this diagram shows a little bit more specifically what we're working on in my lab. So I show this toxin interacting with the human cell, and for one of the toxins that we're working with, that toxin has to bind to both cholesterol and a molecule called LFA1 on the host cell surface. So what we've tried to do is block each of those interactions specifically. So we found that if we block the ability of the toxin to bind to cholesterol, or if we block the ability of the toxin to bind to the protein LFA1, we can prevent the toxin from killing the cells. And this would allow, like I mentioned, the body to clear the infection. We also have a group of projects in my lab where we're looking at these outer membrane vesicles. So these are produced by gram-negative bacteria. I mentioned that gram-negative bacteria have two membranes, and that outer membrane can bleb off to form this vesicle, which is a membrane-encapsulated sphere. It includes toxins and other virulence factors, and it's a way for the bacteria to deliver a lot of molecules at one time to the host cell. So what we're trying to do with this project is understand what are the molecules on the host cell and what are the molecules on the vesicle that allow for the vesicle to be delivered to the host cell. And the idea, again, is that if we know what those molecules are, we could block those specific interactions to prevent disease. We're also looking at how these vesicles are delivered to neighboring bacterial cells. There have been some reports that these vesicles can be used to deliver antibiotic resistance genes to other bacteria. And so, again, we're trying to understand what molecules are present on the bacterial cell that allow this delivery to occur. So I'd like to end by acknowledging my members of my lab. I have a postdoc, 5 graduate students, and 7 undergraduate students. They range from physics, chemical engineering, bioengineering, biological sciences. And I'd like to thank my funding sources. Lehigh has been especially generous in funding my research. And I'd also like to acknowledge funding from the NSF and NIH, as well as Sigma Xi. So thank you.
Great, thank you very much. So cholesterol is a good thing.
It is, yeah. It has a bit of a bad reputation. It's actually a really important component of your cell membrane. So if you don't have cholesterol, your cells aren't going to work properly.
So it's true, there's good and bad cholesterol.
There is good and bad cholesterol, yes.
Yes. Interesting. All right, we have some questions coming in from alumni, so I will read them off to you. Howard Schwartz is asking, do you see any way the business model for private development of new antibiotics can be improved?
Yeah, I think there really has to be some input from the federal government. I think if you look back to the discovery of penicillin in the 30s and 40s, it was discovered in England, and the United States realized the importance of this product, especially as we were starting to enter World War II. And so the government actually invested a lot of money into making this sort of scaling it up so that it could be used by more people. And I think without that type of investment, it's just not going to happen. I think there's a lot of disincentives for the pharmaceutical company to pursue this type of work. It's really expensive to create a new drug, and then antibiotics are unique in that if a new drug were discovered that was really, really good at killing bacteria, it would actually be put on the shelf, most likely, to save for when it was absolutely needed. So it's a lot different than, say, a breakthrough cancer drug where you would be trying to get that drug to as many patients as you could. In the case of an antibiotic, you would actually be holding on to it. And so there's not a lot of possibility of recouping those costs for developing a drug. So I really think it's essential for private foundations and for the government to fund this type of work.
Okay. So Jonathan is asking, do you know how many people are dying currently because we don't have an antibiotic to fight their bacterial illness?
Yeah, so the most recent statistic that I saw from the CDC says that there's 23,000 deaths in the United States every year from antibiotic-resistant infections, which is really high, and I don't think it gets as much media attention as it should.
No,
Yeah.
That's just for the United States?
Just in the United States every year, yeah.
Wow.
Yeah.
All right. So in conjunction with that question, Howard Schwartz is asking how many of those deaths come from adaptation of bacteria that are already present in our society versus those that are brought here by travelers from less developed countries?
So I don't know exactly, but I would bet that most of them come from organisms that are already here. A lot of those infections actually occur in healthcare settings, at hospitals, and I think that it's bacteria that are already here and they're just mutating in those types of settings. And yeah.
So a lot of the deaths are probably happening because people who are already sick
Yes,
are
absolutely.
getting sicker. Basically.
Yeah, I think it's really, I think it's a big problem for everyone, but especially among the immune compromised individuals and people that are just less able to fight these infections naturally. Yeah.
Okay. So Brian Gasta, class of 2000, is asking, can you describe what happens to the bacteria when someone stops their antibiotic course early?
Yeah, so what happens is when you take that antibiotic, all the bacteria are exposed to that particular molecule. The ones that are most susceptible are going to die. The ones that live probably have some sort of way to resist that particular that particular antibiotic. And those are the bacteria that are going to then divide and produce offspring. And those new bacteria are the ones that are going to have that gene to prevent the antibiotic's effectiveness. And so what happens basically when you take an antibiotic but you don't finish it off, you're selecting for the bacteria that are most resistant for that particular drug. And so then the bacteria that are remaining are the ones that are resistant.
Okay. So if I get an antibiotic, I should take the whole thing.
Absolutely. Yes. Yes, take it. Usually it's a 10-day course. Even when you start to feel better, keep taking it.
Yeah. All right.
You want to make sure to kill everything. Yeah.
Are we over prescribing antibiotics, do you think? Is that also causing some of this issue?
Absolutely. I think antibiotics are great and they're really useful when they're needed. I think that doctors do tend to overprescribe antibiotics, and I think that that's also our responsibility as patients to tell doctors, or I guess to not go in asking for an antibiotic, especially if you say have a cold or the flu. Antibiotics don't help. Those are both viral illnesses, and antibiotics are not going to help. So I think recognizing what can and can't be treated with an antibiotic, and also recognizing that, sometimes it's no fun to just wait it out, but sometimes that's the best course of action.
Okay. So colds and flus, wait it out.
Yes, the antibiotic won't do anything anyway.
You're only going to create more resistance
Exactly.
for the rest of us.
Yeah.
Okay. So Jonathan is saying, I understand you have grants from the NIH. Does finding new antibiotics appear currently to be a high priority for them?
I think it is. They have a couple calls for proposals where they seem to be really focusing on this. And there's some private foundations that are also really interested in this problem, which I think that's a great sign. I think it's a really important problem. And like I mentioned, we really need that funding to come up with new approaches to treat it.
Okay. So Nanette Witt is asking an interesting question. Is your lab dangerous in so much as you are working with bacteria? Mm-hmm. And how do you safeguard the scientists working in your lab?
That is a great question. So my answer is yes, our lab is a little bit dangerous. So bacteria are classified as either biosafety level 1, 2, 3, and 4, 4 being the most dangerous, 1 being the least dangerous. My lab is classified as 2. So there are certain safeguards that we have in place that we have to follow these rules. Basically, no eating or drinking in the lab, those sorts of things. How we carry the bacteria, we have to carry it in a specific way and make sure that we're not exposing people. And so, yeah, we absolutely, we follow these guidelines. The students are trained in this type of work, and they're not allowed to work with the bacteria until they're well trained. So my answer is, in a way, yes, it's dangerous in the sense that it's biosafety level 2, but it's not dangerous in the sense that we have safeguards to protect everyone in the lab and outside the lab.
Makes sense. We're all safe. So we have a question from Todd Davies, class of 96. In conjunction with another question that's rolled in. So is the research in your lab targeting any specific group of resistant bacteria, such as, say, MRSA? And also, along those same lines, how well do you believe your target mechanism will address the pathogens of interest identified by the recent CDC reports and the World Health Organization?
So we haven't specifically worked with any of those organisms that are listed by the CDC. I do hope to work with some of those in the future. And I think that a lot of what we're discovering with the organisms we are working with will be translatable. Maybe not exactly, but the same idea. So we're sort of showing proof of principle that this could work, and then it's fairly easily adaptable to other organisms.
Okay, great. So Brian Gazda asks, what is your opinion or recommendation on using antibiotic soaps versus regular soaps for hand washing as it relates to potentially contributing to antibiotic resistance? And I may add, what about hand sanitizer too?
Yeah, I think as much as you can avoid the antibiotic soaps, you should. I think the government recently came out with a recommendation that we all avoid them. I think they're great for people that work in a healthcare setting. In that case, you absolutely would need them. Maybe also, I would say, if you visit a petting zoo, that would be a really good place to use antibiotic hand soaps. But I'd say otherwise, for general use, it's a lot of times it's the friction of washing your hands when you're actually rubbing your hands together that helps to get rid of the germs, and that can be just as effective as an antibacterial soap. Hand sanitizers, I would say, I don't think that would contribute to resistance in the same way, because it's actually a different mechanism. It's usually it's ethanol in there, which just kills the bacteria. So it's not the same antibiotics that you would take, if you had, say, strep throat. I would say they're good for certain situations, but I don't know that you need to sanitize your hands all that often. I think bacteria sound really scary, but in general, most of them are actually really good for you. So, when you kill off all the good bacteria, then it sort of allows the bad bacteria to kind of take over. So I'd say in moderation on the hand sanitizer.
Okay, good. What about things like, I was just thinking about like Lysol wipes. I just wipe my desk down with a Lysol wipe. Is that along the same line?
I would say, yeah, same thing. Same thing as the hand sanitizers. It's a different mechanism, so it's not going to be. It's a really different different molecule, so it's not going to contribute to resistance in the same way. So yeah, I use them too. I'm guilty of it, but again, I'd say in moderation.
All right, great. All right, Howard Schwartz is asking, are there effective strategies for inhibiting the development of resistance by bacteria versus having to modify existing antibiotics or develop new ones?
Yeah, so I don't know that there's any way to prevent resistance from occurring. The thing with bacteria is they divide so quickly. So, bacteria within hours, you have a whole new generation. So if you compare that with humans, the ability for mutations to occur is just so much more likely because there's so many more divisions occurring. So, I think inevitably the bacteria are going to be resistant to whatever we discover, and I don't know that there's any way around that. I think my thought with the research in my lab is that the more tools we have to fight these diseases, the better. So I understand that the bacteria will probably become resistant to the strategies that we're coming up with in my lab as well. At some point, it's going to happen. But if we have more options for treating these diseases, then we're better off.
All right. Along those lines, given the four-wall fortress that you talked about earlier, and the prevalence of problems in lower income populations, which of the four approaches to combating disease are the most cost-effective?
Most cost-effective? I think vaccines are great because you can just prevent diseases from even happening. And then in that case, You don't need antibiotics, and you don't have to worry about these resistance problems. So, ideally, we'd be able to take a shot and be immune to everything for the rest of our lives. I don't know that that's likely. Yeah.
All right. Robert Gordon's saying on several recent cruises. That sounds nice. Yeah. Everyone entering dining rooms has had to use disinfectants on their hands. Do you think this is a good or bad practice? Is it effective? And also, if so, is it done at Lehigh dining facilities?
So I think the reason that they have you do that on cruises is because they have problems with, I know I've heard about norovirus, but I would assume also other infections where, since everyone's in such close proximity, if one person gets sick, everyone's going to get sick, and then that's just a terrible situation. And so I think that the hand sanitizer is for that purpose. I don't think that they do that at Lehigh, and I don't think that they need to, and I wouldn't recommend that they do, because in a cruise ship, again, you're just in that confined environment. At Lehigh, I think it's less of a concern, these types of outbreaks. Although there are different outbreaks that occur.
Sure, they have in the dorms and things.
Yes.
But as long as everyone's washing their hands nicely,
yes.
using friction.
Yes.
All right, good. I think that's probably it for the questions that we have that are coming in. So, all right, so we know take our course of antibiotics, wash our hands.
Yes, yes.
Not
absolutely.
with antibacterial.
Yeah, so. Try to limit it. Yeah, absolutely.
And we'll help the situation.
Absolutely, yeah. I think we all have personal responsibility in there.
All right, great. Well, thank you so much to Professor Angela Brown for joining us today. We really appreciate it. Thanks for sharing your research with us. And for you in the audience, we hope that you will join us on a future Mountain Talk. Thanks for joining us today.